Log on, tune in, find out. Another good idea from Cambridge.
Speaker Val Fedorov will be talking about design of clinical trials with multiple endpoints of different types.
Thank you.
Is it on? Good. Okay, so just let us start. And of course, thank organizing committee, who is Vlad. Are you on organizing committee? Thank you. And all others, I do not know who else, for inviting me. And it's an honor to give a first talk. At least I have an excuse when people fall asleep. It means that probably it's because of jet lag, not because my talk is very boring. So, okay, it's very simple approach which is based on optimal design theory. So, and this optimal design theory consists of a few steps. I think in this particular case, for this particular audience, I selected to dance 8 steps dance, and it will be rather standard approach. Select model, then derive information matrix of a single observation. It will be clear later on what does it mean. Then define utility function. It means define what you want really from your experiment. Then select a penalty function, and the rest you can read on your own. Do you want me to read for you? Okay, Mark. So, but the basic idea is that what I'm trying to do, I try to really use a very, very standard routine technique which is called optimal design theory. So that type of methodology somehow casted for this specific case, clinical trials, or more probably accurately, early clinical trials. And this picture is not mine, so it was kindly provided to me by Peter Ho and Alka Preston during my previous life with GSK. So, and I use this example as motivation. So there is no practical result in my talk, it's just some description of some methods, but they're motivated, or development, all development motivated by this example. So, and you can see that when you have something related to oncology, so usually efficacy of drugs measured by the size of tumor. So it's not actually actual endpoint, but it can be considered as a good biomarker, the size of tumor. And if you start to look at all these pictures, you can see that there are some kind of qualitative description of what is going on, say, disappearance of clinical symptoms or evidence of tumor. So it's called CR, so complete remission. Then there is partial response, and there are some kind of metrics which always were perplexing for me, because they're working either with the area or with the decrease of widest diameter, something like that. And I always was perplexed with this picture because it's a circle, and I thought that circle has one diameter. So, but in any case, you see there are tendency, and its usual tendency, say, to replace some kind of continuous response by dichotomized or categorized response. And people believe that it's simpler, and it's really simpler to talk about. But it's not very good, because obviously we lose some information when we convert some kind of two-dimensional picture in 2 points, yes or no. So it's really will be loss of information. Now, that was about efficacy. As for toxicity, then probably it's even more difficult, because It's so many different types of toxicity, and usually people do not report anything quantitative. They say yes or no, you are vomiting or you are not. You are sleeping or you do not sleep, or whatever. So, and very often they resort even to some kind of common adversity effect that's called SAE, and I try to recall, is it severe adverse effect or okay. It could be viewed as a severe. So usually it's just 1 and 0. And at the very beginning of this story, I think we started it with Vlad Dragalin while he was still at JSK. We decided to play with two-dimensional problem, I mean two responses, and both of them were binary. And what was really important from our point of view was that these responses are correlated. It's not just success or failure, but say success in the sense of efficacy actually depends upon success or failure in the sense of toxicity. So if you are a very weak man and so or woman, so then chances for your cure is much less. So probably something will happen with your kidney or whatever, liver. So there is obvious correlation between success and failure in this. I mean, success in efficacy and failure in toxicity. That's why we try to introduce correlation between efficacy and toxicity, and this binary approach. So it's not that there are a lot of ways how to introduce that, but I think the best and most natural way to introduce this correlation to actually use latent, there are various words for this, latent or background normal distribution. In normal distribution, say two-dimensional normal, everyone can introduce correlation, and then using this latent distribution, normal distribution, you can introduce, so here there is efficacy, here there is toxicity, then you introduce some cutting levels, and if your observation somewhere here, you compute distribution probabilities of corresponding events that you are in this quadrant, and so it's how you use and you create so-called probit model. In this particular case, it's two-dimensional probit model. And here I introduce some notation. So this is latent normal distribution. It's correlated normal distribution. Responses are correlated. And then you can define, for instance, probability that there is no toxicity and no efficacy. A very important event. So there is nothing. So, but nevertheless, it's important to fill all these tables. So you have efficacy, unfortunately toxicity, but you're probably interested when you have efficacy and no toxicity in this probability. Very often I heard that this is very complicated model and not very practical because needs a lot of computation. And I have to say that those who really consider that it's very complicated model, I recommend to read these very good papers recently published. So, and even in those times they knew how to do it. And probably somebody of you heard so-called tetrahoric coefficient of correlation. You have to hear this. Because it was invented by Brits, and so, and in particular, Carl Pearson introduced this tetrahoric coefficient of variation. And it's very popular in psychometric and in econometric. So, but you see, people even in those times considered this very handleable. But it's easy to explain because even in those times, tables of normal distribution already existed. So You don't need to compute any exponent or something. You just use tables of normal distribution. Now, in typical dependence, look at this plot, because we are talking about binary case or dichotomized case. The typical curves are something like this. So probability of efficacy, marginal probability of efficacy going up when concentration, when dose increases, probability of toxicity going up when dose increases, very natural, and probability of efficacy and no toxicity actually has this bell shape. And I do not introduce anything special. Bell shape is very natural if you take into account two responses. It's very natural. Just use formula for calculating of probabilities, and that is all. Now, what is important here? So probably it's important here is that some kind of utility can be described by this function, probability of efficacy and no toxicity. And probably we have to look for those which provide a peak of that function. And it's very natural, and you can report it to anyone, so to your managers, or to FDA, or to EMEA, or to sell your method and think. It's simple and very easy to explain. And Vlad, do you recognize this paper? Yeah. Rather old paper now. Now, but actually we saw that, okay, so it's continuous, but can we do something a little bit better? And so actually, to these two lines, if you do not do the dichotomization, you can find out that these two lines are nothing else but like mapping of two straight lines, but in interval between 0 and 1, because probability could be only in this interval. So, but you still can use, you still can introduce, you see in the framework of this probit model, you can introduce still probability of efficacy defined in old sense, as it was defined in binarization, and no toxicity. I do not use it for any measurements, for data analysis, but I can use this for reporting my data. I just use it for reporting data. For instance, if I find out some person from FDA, and I did, so I can use it for reporting. For analysis, I will use this primitive, line is not primitive, it's very good, I mean straight line, very good object. So, but to report, actually, my function will be still no efficacy and no toxicity. That is for reporting. And if you move further on, then just to emphasize that it's important to work with continuous, to analyze continuous responses. You can report whatever you are asked for. You can negotiate with people who are really looking for your results. So look here, so if I use dichotomized data, my standard error of the best dose is something like 0.05. And if I use continuous data, it's exactly the same. I define the same dose, optimal dose defined in the same way. Then I have this variance 0.03. Oh, it's not variance, it's standard error. So take square of that and find out how many observations or what the ratio of sample size will be. It will be 9/25. So it's quite substantial reduction in sample sizes if you handle continuous data. But I emphasize these two problems are synchronized. It's very often people, when compare continuous and dichotomized approach, they do not synchronize what they are talking about. They talk about some hypothesis testing and very often say, oh, I get better result with dichotomized data. Because of hypotheses are not synchronized. Here we synchronize, and it's easy to synchronize. Now, but because we have two responses, so you can follow now the logic. First it was two responses dichotomized, and dichotomize utility function or whatever, some objective function. Then it was both responses, continuous, dichotomized function, utility function is dichotomized. Of course, I had a PhD student, so I look for a problem, so it will be very natural to have one response is continuous and another dichotomized, and utility function is dichotomized. So we started to do this research just because we have to do something. Lately, I regretted that we started only with 2 responses. If I start with 3 responses, then it could be much more PhD work or thesis is done. But in any case, let's do with these responses of different types. So one response is continuous. So I do not do anything, say, with efficacy. It's continuous. As for toxicity, I say it's 1 when this normal variable is greater than some cutting level, or just 0. We can write down, and those who are interested in this, so it's actually published now, or probably, at least it's accepted for publication in statistical medicine. So maybe it's online. I do not have any more access to statistics in medicine, so so you can find out this paper details. So it's very easy to, relatively easy, to find out likelihood, log likelihood for one single observation. What is one single observation? It's when you take an individual and measure response, efficacy response, and toxicity response. What is unknown parameter? So this is variable describe efficacy. It's continuous. So this variable describe toxicity. So it's defined in this way. It's our unknown parameters. Then there is correlation coefficient and sigma 1, which is variance of our efficacy. And it's obvious that we cannot identify sigma 2 and say eta 2 independently because of dichotomization. So we have 4 parameters to estimate how to do this. And you remember, so for design of experiments, it's only one thing which we really need to start use all machinery known there. It's to build information matrix of one observation. And actually we did it. And we spent probably 2 or 3 days with Ron May, Zhang, trying to to do all these calculations. And then we somehow I decided there are a lot of smart people around, so probably somebody did it. And indeed, we found that it was done in 1595. So people really were interested in very same problem. They were not interested in design, they were interested in estimation, maximum likelihood estimation, and hypothesis testing. But this matrix was calculated. So what we need, information matrix, I called it elemental information matrix because we have some kind of elemental parameters, basic parameters. But you can see that there is still no dependence here upon dose. All this is done some given dose, given mean. So we have to introduce somehow dependence upon dose to, you see here, to show that here there is our line, it's dependence upon efficacy. I think it's black one on the screen. It's actually black. Dark blue on my screen over there. And then toxicity. So to describe this dependence, look only at these 2 curves. To describe this dependence, by the way, how much time I do have? When we start? 25 minutes, okay. So you subtracted a few minutes, okay. Now let's move further on. So to introduce this dependence, you can do it easily. You can say eta 1 depends upon dose, like usual linear regression. And it should not be a straight line. So people usually say, oh, I have to get something bell-shaped. If you want to get some bell shape, so make it polynomial of second order. If you need something fancier, so you need S-shape, introduce, here it's linear model for the sake of simplicity, introduce S-shaped curve, so, and do your exercises. So it could be any, so it's no constraints. And there is a well-known formula how to translate, how to get one information matrix from another one if you do parameter transformation. And this is nothing else as I may transform parameter theta1 to parameter eta1. So I can find out information matrix of single observation, but already in the space of theta1, theta2, and still I have rho. Sorry, I didn't make rho dependent upon those. You can, if you wish. You can make sigma 1 depending upon those. Oh, various generalization are possible, and so if you have some time, so you can do these exercises. And now information matrix is readily available. So what you have now, you see previously we have 4 parameters, because it's 4 basic parameters, mean, probability. Now, dimension of this information matrix of single information matrix will be, of course, M1, it's number of parameters in your regression. So usually it's 2, straight line. Same here, plus M2, so it will be 4. It's what we did. And plus another 2, so 6 unknown parameters. Yeah? You can introduce more if you wish. So, but it's still relatively simple matrix, and actually it's well blocked. So you can see that it's well blocked. And you also can verify that when rho is 0, then we get trivial case when all these responses can be treated, efficacy and toxicity, separately. Now, and unfortunately, what is unfortunately, you can see here dependence upon theta, unknown parameter. Where is sitting? Is actually sitting in these coefficients A0. You can look lately on definition A0, A1, and A2. They depend upon unknown parameters, unfortunately. But there are some kind of constants here. So this matrix, particular matrices, do not contain unknown parameters. Now let's move. Now we have to define utility. And just to be a little bit more innovative, so we decided to introduce utility which was not considered before. And therefore, how we define it? Utility is mean efficacy. It's continuous, so introduce mean value of efficacy, given that there is no toxicity, and multiply it by probability of toxicity. This formula, no toxicity. This formula can be rewritten, so those who know Bayesian formula or formula of multiplication of probability, you can do some extra exercises. So this is utility. And what utility is needed for? Probably I have to define from this utility function the object which I am most interested. And very often it's the maximum of this function. The best dose is the maximum of this function. And after that, I can introduce also penalty. What is penalty? If xv star is my best dose, then, of course, I can introduce a lot of penalty, but I use the simplest one, which was used, for instance, in Robbins-Monroe paper on the procedure, Robbins-Monroe procedure. It's just deviation, quadratic deviation from the best point. And that's what is really important for a patient, particular patient. I emphasize patient. Here there is a multiplier, but usually it will be obvious that everything depends upon ratio of R over C. C is some kind of cost, because every patient, even to NY, even if he is at the best dose, it's still some kind of expenses associated with that. So, and here there are plots of now all curves which we need. I just repeat the same plot. So this is penalty, and you can see that minimum of this penalty is at the best dose, and utility, it's rather flat, by the way. Utility is best dose, toxicity, and efficacy. I emphasize that toxicity and efficacy monotone function, but utility function is unimono. So it's, in this particular case, it's concave, but now let us move further on. So why I bother about this row, by the way, about correlation coefficient? Here I plotted a few functions, not me, of course, it was wrong me, but I asked her to plot. This utility function for different correlation coefficients. And it seems to me it's, on intuitive level, it looks like it's clear that optimal dose depends upon how your toxicity and efficacy correlated. If you're very vulnerable to toxicity, you do not have too much chance to, say, survive or something. So, and you can see that this optimal point is moving when correlation is changing. Vlad, if you like this plot, I will send you. And here is my explanation how to introduce this, what I call penalty or cost function. So what we really have in general, so we have targeted population. It's population for which drug should be developed. So what is a penalty function, or do we need? So probably this targeted population doesn't really care about this penalty function. They care only about this dose should be defined optimal dose with X star as precise as possible, as accurate as possible. So they do not care about what patients really, those who are in trial, experience as individuals. Then there is patient, and for this patient, of course, it's important that if this patient on the dose, which is the best one, so he is a lucky one. If it's not, then sorry, then you have to, not you, but he really suffers, and these sufferings can be described by this. Do you remember x minus x squared, x v star minus x squared? So, but unfortunately, so, and there are designs which try to suit each new patient as much as possible. So I think it's not very good designs. So what we are doing, we try to somehow accommodate all populations in our sample on average. So on average, to make their life easier. And that's why we selected function, which is, you see, average across the sample. So here there are this averaging coefficient. And this phi, capital phi of Xi's design, it can be considered as average normalized toxicity. Oh, sorry, penalty. Now, there is a slide which some of you have seen, actually, it's slightly modified slide which I showed during my previous lecture. But, sorry, Vlad, to use you again, but Vlad approached me after this lecture and said, oh, this slide, Val, is very good. So I decided to show him it the second time, so to make your life easier or more pleasant. So what is a real optimization, actual optimization, say standard optimization problem in experimental design? You have information which you gain. It's described by this function. It's information matrix, some functional of information matrix. And you have constraints that your penalty should be less than capital C. So that is the basic information problem. Actually, this is a discrete problem because n number of patients, it's discrete, But people replace this discrete problem, really approximated by some continuous problem, which looks in the following way. You have to maximize information metrics standardized or divided by normalized toxicity, normalized penalty. Now, just to please Bayesians, there is a Bayesian version of that. And so if you have some prior information which can be expressed in terms of variance-covariance matrix, and subsequently in terms of information matrix, which is inverse in normal case to this variance-covariance matrix, then you can formulate the similar problem, only you will find out that actually the problem is consist in optimization of some amendment to all old information, but also standardized by penalty. And actually, this multiplier is not important. It's on the assumption that we do not pay, I emphasize, we do not pay for information which we get from a priori. And usually Bayesians do not pay. They solicit it or whatever, but usually they get it for free. That's why they are so proud of Bayesian approach. You can get it for free. Now, but because I know that if you, and here there is some kind of information about how to do all this design. So basic idea is you have to build so-called sensitivity function, and then you have to, when you build your design, you have to add point where sensitivity function is approach some maxima, and subtract points where the sensitivity function is below something. And when you have, say, de-optimality criterion, then what you have to do, you have to put more observation where prediction is bad, and remove observation when you know that prediction will be good. And so, and one thing which I want to really emphasize here, if I remove this operation here, removing points, which I can do when I build optimal design on computer, I can add and remove patients at different support points. But if I remove this procedure, then that one is nothing else as a sequential or adaptive procedure of building optimal design. So you add patient, each new patient, to the point where your knowledge of function, your response is the worst, which is equivalent that you can gain the most information here. So on intuitive level, it's very clear. So I want to emphasize that all adaptive procedures which we know in optimal design theory are based on this idea. So here there is the same, but said a little bit more accurately than I just said. Okay, so let me have a short break and now two stages. What I'm promised to discuss two-stage design. So, and two-stage design is quite different from what we call Bayesian approach. If you remember, in Bayesian approach, I have some prior information which correspond in this case to this term for free. I solicited it and I had it. Now, if I have two-stage design, I have to pay for both stages. So first stage, I have to enroll patient. Second, some number of patients, and I have to enroll patients at the second stage. So how to enroll this? So usually what people do, it's in pharmaceutical industry, it was always multi-stage design. So they have phase one, they enroll 20 patients. I just try to do extremes. Then they get some information from 20 patients, and they do next study, which contains 300 patients, thinking that they have enough information from these 20 patients. And then if they follow more, if you have something reasonable after 300, okay, you run with 3,000. So that is partitioning, which you can find out typical in pharmaceutical industry. And Sue Jane can correct me, but on average, the idea is this one. So be cheap at the beginning, and at the end, just invest as much as possible. But what we decided to do, so let us try to figure out what is the relationship between this n0 and n1. So between first, we use here index 0, but it's first stage and second stage. And if you do some arithmetics with optimization problem, you find out that you have to really minimize some linear, you see, information matrix of the total experiment standardized by total expense or by total penalty. And if you do accurately, so you find out that actually optimal design will depend, and so usually xi 0, you select some simple design because you do not know unknown parameters. So you do something simple, say, you take 5 doses more or less uniformly allocated, whatever. So you just talk to your people, I mean, to your colleagues from clinical divisions and select these doses. So it's more or less arbitrary. Of course, you can show them some kind of locally optimal design and so on. But basically, you do not do too much to select this xi 0. And therefore, we are interested mostly in selection, optimal selection of the second stage. Because at the second stage, I will already have some interim estimators. That is important. At the beginning, I have some guesses at the best, if I'm not Bayesian. What the Bayesian have at the very beginning? They have prior mean. Is it or beliefs? You see. They have beliefs. And what is good about beliefs that you cannot test them. You cannot verify. But here you have to, and therefore, let us move further on. So, but it's important to select this pi, which describes partitioning of my trial. So, as soon as I build this sensitivity function, I can apply all tools which were developed in optimal design and try to do some kind of dirty work, I mean, calculations. And so that's what we did. Can you read actually what is on the screen?
No.
Is it my fault or?
To some extent.
Okay. Okay, so then I try to explain. You see, it will take some time. So here there is what? Here there is so-called two-stage plots of histograms of best dose estimators. So for each design, we calculated, repeated many times. So we did Monte Carlo. Repeated many times this fictitious experiment. So we simulated data and then calculated estimated. So this particular case, it was usual two-stage d-optimality. So we have between, say, 0 and 1.4 doses. Why we selected 1.4, I cannot recall. But it could be 0 and 1 or any other boundary. And so this is so-called d-optimal design, no penalties. Just no penalties, standard approach. And in this particular case, by the way, here in this particular case, it's distribution of points where we had to do observation, a distribution of support points. Sorry, I misspelled it previously. It's distribution of support points. And you can see that actually in the sense of penalty or security or safety of your patient, this design is not very good. Because a big number of patients are located at extremes. And on the left extreme, we have no efficacy, it's unethical. And on the very right end, we have also a lot of patients. So it's also unethical because it's very large toxicity there. So now we decide, okay, let us introduce something which is a little bit, let's say, take into account what is going on. And by the way, here it's misprint, so it should be different R. So, and now we introduce penalty, and, oh, no, it's no misprint. So this is distribution of points, this is distribution of best dose. So this is distribution points where measurements were done, this is distribution of estimated endpoint, so this best dose. And you can see that variance here, it's almost unbiased, and variance here is 0.04 for those who cannot read. I mean, because of distance. Now here is the same, but with penalty function. Now penalty function is introduced, this quadratic. Here there is distribution of points where measurements are done. So it's usually 2-point design, optimal design. And you can see that it's distributed quite a bit here, but still we have, sorry, but we have a number of people at toxicity. But that's how we built our penalty function. It was symmetrical, you remember, x minus x with star square. You could do any non-symmetrical function, and then this piece could be minimized. And of course we have, because we have additional constraints, we have larger, actually it's standard error, larger standard deviation. So it's a little bit larger, so it's 0.042, here it's 0.045.
What should be the true best dose in this simulation?
No, it doesn't have, because our function is unimodal.
What is the true?
Oh, if.
What is the true dose in your simulation?
Oh, it's here. 1.
Okay, so you have 6 parameters
Yes.
and you have trinary response.
Bivariate. I mean, 2 responses. One is binary, and
So how do you estimate your 6 parameters with the rank, the matrix of rank 4?
it's not matrix rank 4, but you see there are 2 parameters, which one is sigma, and another is rho, which are related to variability of your data.
Oh, okay.
You see,
I just
when you estimate mu and sigma in normal case, you do observation in 1 point, but you still,
Okay.
Maybe this is a good time to ask that question.
yeah.
Obnoxious.
Are you talking about naive questions?
No, so your slide suggests that what you really care about is you want to find the optimal dose as closely as possible, and you want to avoid putting patients on the excessively toxic dose, right?
Okay, yes.
That was your interpretation.
It was slightly different interpretation. Actually, I do not want to put patients. You see, there is a sensitive, there is a penalty function. There are low doses and high doses.
Oh yeah, that's what you said earlier, but what you said now when you interpreted the slide, you kind of like you show us the standard deviation around the optimal dose in your Monte Carlo, in your simulation, right?
Yes.
And you pointed out it's like, it seems to me the slide suggests narrow standard deviation around the optimal dose is good, right?
No, this is dose. This is dose, how dose was estimated. Here's how point were allocated, how patients were allocated. So it's 2 different things. And what I really try to emphasize when Bася asked, I think, that I don't remember who asked, but you see, because we introduce very primitive function, just this one, so it's symmetrical, either we're moving left towards where there is no efficacy, or we're moving right where there is toxicity. It behaves the same, and it's not very good. From mathematical point of view, for me, it doesn't matter which function. But I probably will introduce something like that. So with efficacy, when I move to the left, it's going like this, and for toxicity, it's going like that. Non-symmetrical. So, but what I'm trying to emphasize, as soon as you quantify your thoughts about efficacy and toxicity, then you just put it in this curve, and then this actually point will be eliminated.
Oh, perfect. So if we get more sophisticated about this curve, then we probably don't need the d-optimal criterion anymore.
Why?
Because you don't care about the parameters really, you only care about the dose.
No, it's only toxicity. But you need to estimate this dose.
That is what this curve formalizes, that you want to estimate this dose x star, right?
Yeah, I want.
But you don't care about the parameters themselves.
Yeah, I don't.
Actually, like one of the parameters is not even identified, right?
Yes, I don't care, but I know that this dose can be defined as soon as I estimated parameters. There are non-parametric methods.
Right, but why the detour? Like,
Your estimator gets worse and worse the further.
But non-parametric, and I will show you a plot. So can you wait a second? Oh, maybe more a little bit. But.
Stephanie might not allow us.
Okay, now let us move, because time is, I'm out of time. But it's not my fault. Now there are two extra plots. Here, by the way, is a design which is very often used in practice and probably what you are talking about. So what if I will do the following? I will do my observation and try to, from this observation, estimate immediately what is the best dose on each stage. How you do this, I do not care. In this particular case, I estimated this dose plugging in parameter estimators in model. You can do it differently. So I called it best intention design. So I place each patient in the dose which is the best accordingly to current knowledge. And then, if you consider that type of experiment, then standard error, standard deviation, is 0.06. You remember previously it was 0.45, something like that. Now it's 0.463, I think. So we lose actually in precision like 6 to 4. And if you square this, it will be 36 to 24. And do cancellation in denominator and numerator. So it's quite substantial difference. So this best intention does not really work here. Doesn't work well. And what is worse actually, if you start to make n's large, asymptotically, this best intention design with probability 1 converge to wrong dose. That is important. Another important message from here, give me 5 minutes, okay? From here is that we also run fully adaptive design. What does it mean, full adaptive? Not two-stage, but full adaptive. We did adaptation after each patient. I consider it stupid because it's logistically very difficult. But we did, mathematically it's nice. And what happens is that actually full adaptive design most often worse than two-stage design. And for those who wants to discuss it, I'm ready to discuss, and there are very solid mathematical explanation of this fact. Two-stage design for nonlinear models can work better or much better than full adaptive design for finite samples. And we can discuss it. Vlad doesn't understand this explanation, but I try to explain him. And here, how to select partitioning. Final slide. So partitioning is the following. So what we did at each point, obviously that if I put all observation at the point, so if I start to design my experiment, build locally optimal design from the very beginning, if I know known parameters, then I will have the best results. And so in this case, I will have this information, this square, empty square, this information. Now, if I do two-stage, I can do two-stage when I still have some observations here. I think it was something like, we started to do simulation with 10, with 20, 50, I do not remember exactly. You can put projections on this axis. So what happens? That we have actually distribution of information which I accumulate at the end of trial. Because second stage was designed having interim estimates only. So obviously I have random situation with some kind of random parameters. So I have to accommodate and the method to this random. And what happens, so here there is plots, these correspond to, because it's random, it cannot be of course better than this, but here there is quantile, I think it's 50%, then 25 and 10. So at least in this particular case, 10% of cases, I can get design which is very bad. When initial stage is very small. And then, of course, variability, as soon as I move in this direction, variability is getting less, but I also get less information because first stage is not designed, and I design only second stage. And when I partition 180 patients in first stage accordingly to bad design, I have only 10 patients allocated accordingly to good design. So that's why there is some kind of interim point, something like here probably, which somehow a good compromise between probability of bad designs and probability of good design. So it will be this point. And whatever we did, usually the ratio of that type of exercise, in that type of exercise, you have to place something about the 30% or 40% of patients at initial design. Of course, it's mathematics, I can do it the other way around. And about, say, 60% of patients to the second stage. But it's not like 30 and 300. It's what usually people do. They always say, oh, I have a method which allows me to do everything with 30 patients. And then drug fails, maybe even good drug. Now, you have summary, so you can read, and I open to question, but we have no time, so it's good.
Okay, we already had a lot of discussion and questions, so let's have one quick question. Ben?
You could use other distributions than the multivariate normal. What about copular, which might...
Oh, you can do a lot, and actually in the first paper we didn't use a normal distribution, so we use slightly different approach. So it was, you see, you can do whatever you wish, but normal distribution, it's a very natural way to introduce correlation. But of course you can introduce any elliptical distribution, which is also easy to introduce correlation. Or it's up to you. The idea is that it's actually to use some background distribution or latent distribution to get feeling of some kind of what is going on.
Yeah, actually a related question that if you have a dichotomized outcome, then why not use Dirichlet distribution? You know, because if you use the bivariate normal and try to cut it, it's still constrained, right? I mean, it's not totally unconstrained. So why not use the more general Dirichlet distribution?
But you can, but it's more difficult to introduce correlation which you can trace in Dirichlet distribution. As soon as you introduce normal distribution, it's easy for me to explain all correlations. But it could be, you see, it's 2 by 2 table. So you can introduce correlation implicitly or explicitly, doesn't matter. This is a convenient tool when I can merge different types of variables. What is important about this approach, when I have latent normal, I can include cardinal variables, I can include, you see, binary, I can include continuous, whatever you wish.
Could I make a comment in that regard? We used a more general version of this for multivariate ordinal toxicities in a paper with Bacalla about 8 years ago. And I can attest to the fact that it's wonderfully trackable, that you can do many, many simulations, and the MCMC runs beautifully. So this sort of model is extremely tractable.
Which one?
with using latent variables to account for discrete variables, latent variables in the computation.
Easy, that's what I'm trying to say.
It's a great tool.
It was certified in century ago.
Okay, given the time, I suggest we stop questions.